here.
Good morning, everyone. We were talking about citric acid cycle last time we met. I told you that it holds a central position in aerobic metabolism. And we talked about the acetyl-CoA that is channeled to the citric acid cycle as it can stem from degradation of carbohydrates. So we also talked about the pyruvate dehydrogenase complex reaction, where pyruvate is actually broken down, partially to release one carbon unit in the form of carbon dioxide, and the other two carbons are then linked as acetic acid to coenzyme A to form acetyl-CoA. But then I also indicated that the acetyl-CoA that is channeled to the citric acid cycle can also stem from degradation of amino acids, and with this, of course, peptides and proteins, and fatty acids which stem from lipid degradation. So what we're going to do today is we'll talk a little bit more about the citric acid cycle. We'll go over the 8 reactions that make the cyclic pathway. We'll talk a little bit about regulation and then the so-called anabolic nature of the citric acid cycle. Before then, we will continue Thursday or next week, actually, I guess, talking about degradation of fatty acids, which will be followed by a discussion of degradation of proteins and amino acids. So as I did with carbohydrates, I will also provide you some background information, again, as it is related to lipids and also proteins. And only after we have talked about degradation of proteins and lipids, we will then talk about what happens actually with the electron carriers NADH and FADH2 when energy that is stored in these molecules is utilized for ATP production. This is coupled to cellular respiration. So today our focus will be on the citric acid cycle, and I gave you a little bit of more or less general information last time we met. Told you it's made of 8 steps. It's a cyclic pathway. So one of the starting materials of the first step is actually regenerated in the last step. In this case, it's oxaloacetate, a 4-carbon dicarboxylic acid. I talked a little bit about nomenclature. I encourage you to familiarize yourself with the names of the simpler dicarboxylic acids because there are a number of dicarboxylic acids found in this pathway, despite the fact that it's called tricarboxylic acid cycle, or perhaps cycle, or citric acid cycle. So do yourself a favor and look at these names again. I also pointed out that there are 2 steps actually where carbon dioxide is released. So 2 carbons, 1 2-carbon unit actually enters the cyclic pathway in the form of the acetyl group coming from acetyl-CoA, and then 2 1-carbon units are released in the form of carbon dioxide. So again, in a stoichiometric sense, the 2 carbons that enter the cycle actually are removed. Now looking at these red boxes here that show where the carbons actually end up, it's not the very same carbons in the first round that enter the cycle and then are released, but from a stoichiometric point of view, 2 carbons enter and 2 carbons leave the cycle. Okay. Then there are 3 steps where NADH is produced. Again, when you see NADH, think energy also. And then there's 1 step where FADH2 is produced. Again, think energy. And then we have 1 step where an ATP equivalent, so either ATP itself or GTP is produced. So overall, just going through the cycle 1 time produces a lot of energy carriers. So it's very important for aerobic metabolism. So as we did for glycolysis, we will take a look at the chemistry of these individual steps, and we'll take a look at some select enzyme mechanisms to understand how these enzymes work. And then again, we'll take a look at energetics briefly and regulation. So the first step is the transfer of the 2-carbon unit, the acetic acid part of acetyl-CoA, our 4-carbon molecule oxaloacetate. So again, nomenclature, oxalo comes from oxalic acid, would be a 2-carbon dicarboxylic acid. Here we have a derivative where we have it linked to acetic acid, okay, making this then dicarboxylic acid, which in alpha position to one of the carboxyl groups has a ketone. Okay? So we transfer a 2-carbon unit to the 4-carbon unit to make a 6-carbon tricarboxylic acid called citrate. So that's the ionized form, or if you protonated this, it would be called citric acid. So citric acid is our first tricarboxylic acid, meaning we have 3 carboxyl groups linked to this molecule. And then we have in central position here hydroxyl group. So this reaction, which we're going to take a look at in more detail in a moment, is synthesized, is catalyzed by citrate synthase. And as you can see here from the energetics, this is a rather hexagonic reaction, both under standard transformed, meaning laboratory conditions, but also under physiological conditions. And it's our first regulated step within the cycle. So when we will talk about regulation of the citric acid cycle, we will also take into account the regulation of the pyruvate dehydrogenase complex, which is of importance when our source of carbons, the acetyl-CoA, actually stems from degradation of carbohydrates. But within the cycle itself, we have 3 regulatory steps, and this is the first step. So this is an irreversible reaction, also under physiological conditions, catalyzed by this enzyme called citrate synthase. So the mechanism of this enzymatic reaction is shown here. We have oxaloacetate shown here in blue. We have our acetyl group of the acetyl-CoA shown in red here. We see that we have a histidine residue in the active side of the enzyme, and we have an aspartate residue in the active side. And this is a classical acid-base catalysis. So what happens is that a beta nucleophile is produced by abstracting a proton here with the help of this aspartate residue to form a carb anion, which we can also formulate as an enol. Okay, so we abstract the proton here, these electrons can move over here, and then we have this structure stabilized through the formation of a hydrogen bond. Now this is named here a low barrier hydrogen bond. This means that it's a very stable bond, and the distance here between our atoms that participate in this type of hydrogen bond are very short, and it's pretty much under X-ray crystallographic pictures. It's actually very difficult to determine whether this would be a covalent bond or non-covalent bond, and whether the hydrogen would actually be equally shared by these 2 electron withdrawing nitrogen and oxygen respectively. So this is a very stable bond. Nevertheless, the acetyl group then attacks our partially positive carbonyl carbon oxaloacetate. I pointed out that we have a ketone here, carbon number 2 of the alpha carbon with respect to one of the carboxyl groups. So we have an attack here. These electrons move over here with the help of a histidine residue. Here we protonate this oxygen then to form the hydroxyl group that we will find in citrate. Okay, and we have at this point formed a derivative of citric acid, which is still covalently linked to coenzyme A. Okay, so again, remember our 2-carbon unit, our acetyl functionality, our acetyl group comes linked to coenzyme A. We have this formation of the bond, and at this point, our intermediate is still linked to coenzyme A, so we have a derivative of coenzyme A that is linked to citric acid. Now at this point, our citric acid part is chiral, because this is a chiral center, because this carbon is linked to 4 different substituents. We have the hydroxyl group, we have the carboxyl group, we have this acetic acid group, excuse me, and then we have our coenzyme A linked to these 2 carbons still. Okay? So at this point, this is a chiral molecule, and the chirality is such that this is the S enantiomer. Now, after we hydrolyze this bond between our citric acid and the coenzyme A, we release coenzyme A and we form our product, citric acid or citrate, because pKa's of these carboxyl groups, under physiological conditions, they will be deprotonated, so it's appropriate to call it citrate to indicate that it's deprotonated. You may remember that when I talked about formation of ethanol, I talked a little bit about prochirality. Okay, so also here in this case, we have a prochiral molecule citrate, where the enzyme could actually distinguish between this arm, which is called pro-R arm, and this arm called pro-S arm. So in reality, this is not a chiral molecule, but if we substituted one of these arms here with something else, we would form a chiral molecule. And if we just link this molecule here again to coenzyme A, we would form this chiral molecule. So remember that this concept of prochirality is something that is historically related and most appropriate in the context of enzymology, because enzymes can actually distinguish often enough these 2 sides in a molecule that is not yet chiral and can make them chiral. Okay, so the same applies here to citrate as it applied to ethanol. The enzyme that catalyzes this reaction has a molecular weight of about 100 kilodaltons. It's a dimer, here we see the 2 dimers, and also undergoes a significant conformational change, a so-called induced fit upon binding to oxaloacetate, and this induced fit then opens up a binding site for the acetyl-CoA, and then we have the reaction occur. Okay, so at this point we have formed citric acid, citrate, which in the second reaction is converted into another tricarboxylic acid, isocitrate. As the name implies, it's an isomer of citrate. Okay, and if you compare these 2 structures, the one where these 2 moieties are shown in red boxes with the one where they are shown in blue boxes, we see that the positions of the hydrogen and the hydroxyl group respectively have changed. Okay, been switched. So we have converted citrate to isocitrate. Now this is de facto an isomerization reaction, but based on the fact that this isomerization is achieved by first dehydrating citrate, so removing a water molecule in the form of a hydroxide ion and a proton, and then rehydrating our intermediate here, which has a double bond, it's called cis-aconidate, we can actually classify this enzyme called conitase as a lyase. Okay, so this is not the strictest sense if we look at the type of chemistry this enzyme performs. It's not an isomerase or simply isomerase, but it's a lyase because it also removes functional groups, and this leads to the production of a double bond. Okay, and you may remember that we had the same situation when we talked about glycolysis and talked about the enolase step, where I said that we have here also a lyase. This wasn't really an isomerization reaction, but this was the first type of lyase reaction that we saw actually talking about metabolism. And here we see it again. And despite the fact that you may think, okay, it's an isomerase that catalyzes this reaction, what is decisive here in the nomenclature is what type of chemistry these enzymes perform. A conitase, obviously this is a reversible reaction. A conitase is actually an interesting enzyme for a variety of reasons, not only because it's a lyase, okay, if you find this interesting or not, it's up to you, but it's actually an iron sulfur protein, which means it has a so-called iron-sulfur cluster. And we will see a couple of iron-sulfur clusters when we talk about the electron transport chain. In the electron transport chain, these iron-sulfur clusters actually help the proteins that have these iron-sulfur clusters to transfer electrons. In the case of a conitase, the iron-sulfur cluster plays a different role, namely to hold the substrate in place. Okay, so there are different types of iron-sulfur clusters. Often enough, they are relatively difficult to characterize and analyze structurally. For example, in the case of the electron transport chain proteins that have these iron-sulfur clusters, we're talking about membrane proteins. That's not the case here for a conitase. It's somewhat simpler to characterize. But again, there are different types of iron-sulfur clusters. In the case of a conitase, we have a so-called 4-iron 4-sulfur cluster, where we form a cubic structure that is made of 4 ions and 4 sulfides. And this cubic structure is then held in place in the active site of the enzyme, outlined here in blue, through the interaction with appropriate amino acid residue. Now, in the case of a conitase, these are all cysteine residues, and cysteine residues are the most common amino acid residues to interact with iron-sulfur clusters. When we talk about the electron transport chain, we'll see at least one example where also a histidine residue actually participates in this kind of interaction, but typically it's cysteine residues. And we see here that this iron-sulfur cluster here interacts with our substrate of the conitase reaction, citrate. The mechanism is shown on the previous slide. I would like to go over this real quick because it's also relatively interesting. But we'll see that, okay, the iron-sulfur cluster doesn't really do a whole lot but interact with the substrate and stabilize the substrate in the intermediate. Okay. Now our substrate here is citrate, shown here. Okay, a little bit complex, this picture. We have a couple of amino acid residues coming from the active site of the enzyme: a serine, arginine, aspartate, histidine. What is important here is to realize what kind of reaction we are seeing here. It's a dehydration reaction. So what we have to do in the first step of the enzymatic reaction is we have to remove a hydroxide ion and a proton from one carbon, hydroxide ion from the other carbon. Hydroxide and proton stoichiometrically make a water molecule, so we have removed a water molecule, and we form a double bond in the intermediate. And then we just add a hydroxide ion again and a proton again, but to the other carbons to rehydrate the molecule. Okay, so what we have to do is we have to look for appropriate amino acid residues that can actually help us remove a hydroxide ion and a proton respectively. Now, in the case of the proton, it's relatively simple. There are a variety of amino acid residues that could help us with this. In the case of a conitase, it's a serine residue here, deprotonated alkylated serine that abstracts a proton here from carbon number 2. These electrons move over here, and then at the same time, we actually have probably a protonation of the hydroxide. So if hydroxide is removed, to the best of my knowledge, often enough an amino acid residue in the active site of the enzyme actually donates a proton to convert the removed hydroxide ion immediately into a water molecule. This simplifies matters to the best of my knowledge, and here actually donates a proton. We'll see other cases where other amino acid residues, often enough glutamate residues, donate a proton in such reactions, or sometimes also aspartate residue, I suppose, but often enough it's glutamate residues. So, protonate this hydroxide, remove the hydroxide, and we form a water molecule, okay, and a protonated serine residue. And then the way this reaction is typically depicted, we have a conformational change, and to make drawing these reactions easier, it's usually shown as a flip of the substrate, of the intermediate in this case. So to make things easier, now we just flip our intermediate so that the carbon number 2 that was before at the bottom is now on the top again. What this conformational change actually looks like in reality is a different story, but in textbooks usually you see this kind of flip, okay? Just to illustrate also that then we reverse the dehydration to perform the rehydration, and we simply add a proton now at carbon number 3. Remember we removed it from carbon number 2 before, and add a hydroxide ion to carbon number 2. Remember we removed it before from carbon number 3. The cis aconitate, so this is cis double bond here, and form isocitrate. Now isocitrate here is a chiral molecule. Okay, we actually have two chiral centers, carbon 2 and carbon 3. Carbon 2 here and carbon 3 here. And the stereochemistry is such that we have the R configuration at carbon number 2, and the S is that we go from an achiral molecule to a chiral molecule. I said that aconitase is quite interesting. If it's just for the fact that it's an iron sulfur protein, but it's also interesting because it also has different functions. So there are two different isoforms of this enzyme. One that occurs in the mitochondrial matrix and participates in the citric acid cycle. Remember, the citric acid cycle actually occurs in the mitochondrial matrix. Okay, and then there is also a cytosolic variant of this enzyme. This cytosolic enzyme actually is important for iron homeostasis. So our cells have to have a certain concentration of iron because there are a number of iron-containing proteins, and the aconitase actually is important in regulating the uptake and storage of iron in cells. And the way it does it is that when iron concentrations go down, this iron-sulfur complex actually dissociates, and the apoprotein, so the apoprotein that lacks here our cofactor, which would be the iron-sulfur cluster, then actually can interact with mRNAs and lead to an increased production of transferrin. So transferrin is the protein that transports iron in the bloodstream, and also it results in an upregulation of the production of the receptor that binds to transferrin, and it also leads to a let's say reduced production of ferritin, which stores iron. Okay, so we have more transport and more release of free iron in the cells, and in this way can increase intracellular iron concentrations. Okay. So our enzyme here, the protein aconitase, has two very different functions, and when we have such a situation that the same enzyme protein actually fulfills very different functions, we can call this moonlighting. So there's a little bit more information about this in your textbook if you're interested. And then the cytosolic enzyme actually is also important here to convert citrate into isocitrate and vice versa. Okay, so and this is coupled also to the next reaction. Talk a little bit more about this in a moment. So for the time being, we focus on citric acid cycle. Okay, we have converted citrate. It was from the first reaction to isocitrate. And in the third reaction, the isocitrate, remember, still has 6 carbons, is decarboxylated in oxidative decarboxylation. We talked about oxidative decarboxylation when we talked about the pyruvate dehydrogenase complex reaction, where we also removed one carbon unit in the form of carbon dioxide and actually had a redox reaction overall that led to the production of NADH. Now here we have another oxidative decarboxylation. So our 6-carbon compound isocitrate is actually converted to a 5-carbon compound. Shown here, it's called alpha ketobutyrate. and one carbon dioxide that is released. Let's first talk about the nomenclature again. So isocitrate we've discussed. Alpha ketoglutarate is a dicarboxylic acid. There's two carboxyl groups. It's a dicarboxylic acid that has 5 carbons. Okay, so the dicarboxylic acid that has 5 carbons would be called glutaric acid, or the ionized form is glutarate. But this glutarate here actually has in the alpha position, so in carbon number 2, with respect to one of the carboxyl groups, a ketone. And therefore, we can refer to this molecule as alpha ketoglutarate. Okay? The oxidative decarboxylation is catalyzed by an enzyme called isocitrate dehydrogenase. And the isoform, the isozyme that is found in the mitochondrial matrix, actually utilizes NAD+ as a cofactor, which is reduced to NADH. This is actually an hexagonic reaction, so these two arrows are a little misleading, I guess. Also under physiological conditions. And this reaction is our second regulated step within the citric acid cycle. Okay. Now, you see that in parentheses, shown here on the slide is also the possibility that the enzyme uses NADP+ for this reaction. Now again, this is a little bit misleading, but this is true for the cytosolic isozyme. Okay? So there is also a cytosolic form of the enzyme, and the cytosolic form uses NADP+ and converts it to NADPH. Now I may or may not have mentioned this before, probably not yet, but what I've mentioned actually is that overall NADPH is utilized in anabolic reactions. You may remember that, while NADH is produced in catabolic reactions. Okay, that's a general rule of thumb. So NADPH is extremely important for anabolism. However, there are only relatively few reactions that produce NADPH. Okay, and this reaction is one of the few reactions that produce NADPH. I'll mention a couple more. So this is actually relatively important in that sense. The reaction is shown on this slide and shows us why we actually have this kind of redox reaction here, which results in a decarboxylation. Because if you look at the structure of isocitrate, we see that with respect to this carboxyl group that is going to be eliminated, we introduce a carbonyl here at the so-called beta carbon. So let's take it step by step. I talked about alpha keto, for example, before. So whenever we utilize this nomenclature where we indicate a position by alpha, beta, gamma or so, we refer to a particular functional group. Okay, so in this case, we refer the position or relate the position to a carboxyl group. So before we had alpha, or here we have alpha ketoglutarate. So the alpha is the first carbon following our carboxyl group here. De facto, it's carbon number 2, but in this alpha beta nomenclature, it's actually the second one. Now, if you look at the substrate here for this reaction, isocitrate, if we focus on this carboxyl group here, which is going to be eliminated in the form of carbon dioxide, this would be the alpha carbon, and this would be the beta carbon, right? So in beta position, here we have a hydroxyl group, and this hydroxyl group is being oxidized in this redox reaction, where we produce NADH, and it's oxidized to a ketone. Okay. Now what you have to know here is that beta keto acids are not particularly stable, and for that reason, it's easier actually to kick out this carboxyl group following the oxidation of the hydroxyl to a ketone. Okay, so then we kick out the carboxyl group, and after some rearrangements here, we actually form alpha-ketobutyrate. Okay. So this oxidation actually helps us to kick out the other carbon dioxide. At this point, we have a dicarboxylic acid, alpha-ketobutyrate, which in the next step is also relieved of one carbon unit in the form of carbon dioxide. So we have another oxidative decarboxylation to reduce our carbon numbers from 5 to 4. And we form a derivative of the 4-carbon dicarboxylic acid, succinic acid, or the ionized form succinate. And our derivative is actually linked to coenzyme A. And therefore we can refer to this molecule as succinyl-CoA. So we have 4 carbons, 1, 2, 3, 4. Instead of having a second carboxylic acid, we have linked this carboxylic acid to our thiol group in coenzyme A to form a thioester, okay? And we form succinyl-CoA. So it's an analog of acetyl-CoA that we use, for example, in the pyruvate dehydrogenase complex reaction. But here our carboxylic acid unit is not acetic acid, but it's succinic acid, and it's linked to coenzyme A, and therefore we call this product succinyl-CoA. Interestingly enough, the enzyme complex that catalyzes this reaction, the conversion of alpha-ketobutyrate to succinyl-CoA, is very similar to the pyruvate dehydrogenase complex. They are evolutionary related, okay? So we have pretty much the same chemistry occur here in this oxidative decarboxylation happening to alpha-ketobutyrate as we discussed it in more detail for the degradation of pyruvate to acetyl-CoA. Okay, remember that? We had 3 enzymatic activities. We had 5 cofactors participating in this reaction. Same is true here for this reaction that is catalyzed by the alpha-ketobutyrate dehydrogenase complex. Okay, so not the pyruvate dehydrogenase complex, which utilizes pyruvate as a substrate, but the alpha-ketobutyrate dehydrogenase complex, which utilizes alpha-ketobutyrate. With respect to the chemistry, and I encourage you to actually draw this out, maybe, it's analogous. Okay, but instead of having the decarboxylation of pyruvate occur with the help of thiamine pyrophosphate, we have decarboxylation of alpha-ketobutyrate occur. Okay, then we have a succinyl residue that is transferred here to coenzyme A, but otherwise the remaining steps are actually the same. So if we look at the 3 enzymatic activities that form this complex here, and again there are multiple copies of this also that make this huge complex. The first one, E1, is alpha-ketobutyrate dehydrogenase. Okay, so here we can use the same enzyme that we use in the pyruvate dehydrogenase complex. Then instead of transferring an acetyl residue, which we had in the pyruvate dehydrogenase complex, here we transfer a succinate residue, so we also need another E2 dihydrolipoyl transsuccinylase. But then our third enzymatic activity, which regenerates here our lipoic acid residue, which we need in the redox reaction, you may remember that, is actually the same. Okay, we can use that. I mean, it's not in the very same protein complex, but it's the same protein. Okay, and it's called dihydrolipoyl dehydrogenase. This reaction is hexagonic and is regulated. So this is our third regulatory step within our citric acid cycle. So at this point, we have formed succinyl-CoA. And what you may remember from previous discussions is that thioesters are energy rich. You may remember our discussion of bioenergetics, that there are energy rich phosphorus or phosphate containing compounds, okay, and hydrides are energy rich. We talked about this. Phosphor and hydrides, of course. But then I also mentioned, and you could see in at least one of these tables, that there was also a thioester, and I think it was acetyl-CoA listed, and I mentioned that thioesters are also energy rich, have about the same energy content as one of the phosphate and hydride bonds in ATP. So this energy here that is stored in succinyl-CoA is, as a matter of fact, utilized in the next step of the citric acid cycle, the fifth reaction, to produce an ATP equivalent. Okay, now talk about what I mean by that in a moment. But what happens is that we form an ATP equivalent, either ATP or GTP, and cleave this bond here between our succinate portion and coenzyme A and release succinate, our dicarboxylic acid that has 4 carbons. With regard to the nomenclature, this is a reversible reaction. The enzyme that catalyzes this reaction is called, so it's not meant to be a synthase, okay, but synthase is just synthesize materials, stuff, okay. Synthetases also synthesize materials, but under ATP consumption, okay. So this enzyme here, called succinyl-CoA synthetase, is actually named for the reverse reaction, namely the production of succinyl-CoA utilizing succinate and consuming an ATP equivalent. Okay, so synthetase indicates ATP consumption or ATP equivalent consumption. So I said ATP equivalent, either ATP or GTP. In at least older versions of biochemistry textbooks, you may still find this in newer versions, depending on the textbook, you will see that it's believed, or was believed at least, that in mammals and humans, this reaction produces GTP, okay, and not ATP. While in plants and bacteria, this reaction was supposed to only produce primarily ATP. Now there are actually 2 isoforms of this enzyme, and one can produce GTP and one can produce ATP also in humans. Okay. So you may want to take what you see in textbooks with a grain of salt, but as a matter of fact, it doesn't really matter whether in this reaction GTP or ATP is being produced, because these are readily interconvertible. So we have a GTP, can easily be converted to an ATP. If we have an ATP, we can easily convert it into a GTP. So from an energetic point of view, it doesn't really matter. And therefore, we refer to these molecules as ATP equivalents. With regard to the reaction mechanism, so we want to form ATP or GTP, doesn't really matter. I told you that we have an energy. What happens is that this bond here between the succinate and the coenzyme A is not simply hydrolyzed, because this will just lose the energy stored in this thioester bond. But what happens is that we have this kind of phosphorylasis again, where phosphate comes in and actually cleaves the bond, and we form a phosphorylated succinate, which we can call succinyl phosphate. So this is what kind of functional group here that we have? Succinyl phosphate. What do we have there?
Mixed anhydride.
What's that?
Mixed anhydride.
Mixed anhydride, also called acyl phosphate. And what do we know about this?
It's energy rich.
It's energy rich. So we have not lost the energy, okay? We have still energy in this mixed anhydride. Then from succinyl phosphate, the phosphate is actually donated to a histidine residue in the active site of the enzyme to form a phosphorylated histidine, which is also energy rich, supposedly. And from here, the phosphate is actually transferred to either GDP or ADP to form either GTP or ATP. Okay, so it's a 3-step, multi-step reaction actually to transfer then a phosphate to ADP or GDP. Okay, so at this point we have a 4-carbon dicarboxylic acid, succinate, and succinate is converted to fumarate. So what kind of reaction do we have here? We introduce a double bond here. What kind of reaction is that? you see something highlighted in red boxes, this should help you find the answer. Come on. The title tells you it's oxidation of succinate to fumarate. Why should you see that this is an oxidation? Yes, we have a dehydrogenation. Okay, dehydrogenation, I told you, is equivalent with an oxidation accident. Okay, so this is an oxidation. So we only have redox, coupled redox reactions. So if this is our oxidation, where we have a dehydrogenation, we remove two hydrogens here. We have to have a reduction also, and what is being reduced is flavin adenine dinucleotide. And we saw the structure previously when we talked about the pyruvate dehydrogenase complex reaction. And FAD is converted to FADH2, so the reduced form of this cofactor. The enzyme that catalyzes this reaction is also interesting. Here it's called succinate dehydrogenase, very appropriate. Okay, it's an oxidoreductase, utilizes FAD, we can call it dehydrogenase. It's interesting also because this enzyme is identical with the protein complex that later, when we talk about the electron transport chain, I will usually refer to as complex 2. So you may have heard before, I will tell you when we talk about the electron transport chain that there are a couple of protein complexes that participate in electron transfer in cellular respiration, and complex 2 actually is this very enzyme here that participates in the citric acid cycle. And the electrons that are actually taken away with these two hydrogen atoms, okay, are transferred first to FAD. So succinate dehydrogenase is a flavoprotein, contains this flavin adenine dinucleotide. And the electrons that then are linked momentarily to the flavin adenine dinucleotide are then transferred to other components of the electron transport chain, and ultimately to oxygen, which is the ultimate electron acceptor in the electron transport chain. Okay, so here electrons are taken away here from our succinate, our substrate for this reaction, but then they are donated further down to other components of the electron transport chain. Okay, so when you look at metabolic pathways, I mean, for you it's probably too early yet, but let's say once we talk about the electron transport chain, try to also see some relationships between different pathways. And here we have an enzyme that you may think of, oh, it's an enzyme of the citric acid cycle, but it actually has a much bigger role also if you look at the big picture here, because it participates directly in electron transport also. As indicated by these two arrows here, and the energetics is a reversible reaction, okay? And we'll talk about this protein in a bit more detail when we talk about the electron transport chain. For the time being, notice that the product here of this dehydrogenation reaction is a trans acid, fumaric acid, or the ionized form fumarate. So this is a trans double bond, and as is typical for enzymes that deal with chiral molecules or, let's say, certain stereoisomeric forms, the enzyme is stereoselective here also. It produces, in this reaction as written, the trans acid and would utilize the trans acid in the reverse reaction, but it does not produce or accept the cis acid, which is maleic acid, or the ionized form is maleate. Okay. I have to warn you about these metabolites starting with an N. There are a couple that sound very similar. So here we're talking about maleic acid. A little later I will talk about malic acid, and there's something that is, I mentioned already, malonic acid, so don't confuse these. This is maleic acid. Fumarate in the next reaction, there are only two left to complete the citric acid cycle is hydrated. Okay, should know by now what a hydration is, what a dehydration, rehydration is, and such. But here we have a hydration, meaning we add a proton and a hydroxide ion to the double bond. So effectively we add water, but not as a water molecule, but separately as a hydroxide ion that we add to one carbon that participates in the double bond, and a proton that proton to the other carbon that participates in the double bond. So the enzyme that catalyzes this hydration is called fumarase, or another name that makes this reaction clearer is fumarate hydratase, because it hydrates fumarate. So we add a hydroxide ion here, form a hydroxyl group, add a proton here, form a hydrogen, and form this current molecule that is called malate. Okay, so I just mentioned that you should be cautious about these metabolites starting with M. This is malate, not maleate. Okay, I didn't come up with this nomenclature, so I'm plain B, but be careful. Okay, here and this would be malate, or malic acid. Okay, we have the protonate form. It's a reversible reaction. The change in free energy is close enough to equilibrium that we can assume that this is a reversible reaction. It is both under experimental conditions and also under physiological conditions. Now, in textbooks, what you find is are two possibilities how the hydration can occur theoretically. So you could either first have a proton added to the double bond, go through a carbocation ion, and then add a hydroxide ion, or first add hydroxide ion to the double bond, form a carb N ion, and then add the proton. At the end of the day, it doesn't really happen in which order it occurs. According to your textbook, this variant, I believe, is more likely, but it doesn't really matter. Okay, you have addition of hydroxide ion, the proton, and you have a hydration. And you have formation of malate, which in the next step is oxidized, as the title implies, to oxaloacetate. Remember, this was one of the starting materials in the first step, citrate synthase reaction. So why should you see that this is an oxidation reaction? Tell me two things. Why, even if you didn't have the title here, you would see as a biochemist immediately that this has to be a redox reaction. Yes, you convert the hydroxyl group to a ketone. Okay? We should know that this is an oxidation reaction. What else? If we look at the whole reaction here, what else has to tell us that this is a redox reaction? Keep it simple.
You're also reducing your NAD plus to NADH.
Yes, exactly. We're converting NAD plus to NADH, the oxidized form of the cofactor to reduced form. Now, if we want to continue like that, what else, if you look at this whole reaction as shown here, just disregard the title, what else tells us that this is a redox reaction? Here we go. If you see a reaction written like this, and I would ask you, give me 3 reasons why this is a redox reaction. And look at the name of the enzyme. It's a dehydrogenase. Okay. They catalyze redox reactions. Okay. Then you could start, if you really wanted, you could start counting oxidation states of carbons and things like this also. Okay. So, as a biochemist, having been in this class for 6 weeks now, you should see these things. Okay. I preach this over and over. You may be overwhelmed by the complexity of this material. You have to boil it down to the essentials. Okay? So think about what I told you before when we talked about bioenergetics. Dehydrogenation, hydrogenation, If we have something like this, we're dealing with redox reaction. We have talked about our cofactors, NAD plus and FAD, which can be converted to NADH and FADH2 respectively, participating in redox reactions, and the enzymes, the reductases we refer to as dehydrogenases. These are all things that should immediately tell you that this has to be a redox reaction. Okay. So if you have to find your way around, look for things like that. Okay. Okay, so not quite done yet. So we produce NADH, and this is the third step within the citric acid cycle where NADH is produced. And again, when you see NADH, think energy. There are 3 molecules of NADH produced, as we'll see in this overview also. Here, we have 1 molecule of FADH2 produced, okay? Also think energy, and then we have an ATP equivalent produced. Also remember, we channel 2 carbons in the form of 2 carbon unit in the form of the acyl group from acetyl-CoA, and we kick out 2 1 carbon units in the form of carbon dioxide. Any questions so far? Then let's talk about how the citric acid cycle is regulated. So regulation of citric acid cycle is very important because it has such a great importance for metabolism, and it's pretty tightly regulated also. Now, it's regulated at various levels, and if we look at carbohydrates being the source of acetyl-CoA that is channeled into the citric acid cycle, we also have to take into account regulation of pyruvate dehydrogenase complex, as I said before. So let's talk about this first. And it has been found that the pyruvate dehydrogenase complex is modulated both by a bunch of allosteric effectors, and most important ones are shown here. I'll go through them in a moment. But then it's also been found that the pyruvate dehydrogenase complex is also subject to transient covalent modification. Okay. And so the activity can be modulated based on the fact, we'll see this in a moment. But let's first talk about the allosteric effectors that are shown here. So there are a couple of allosteric effectors that obviously deactivate the complex or reduce the activity. When we say deactivate, we always think, okay, we knock out activity completely, but often enough it's just down regulation of the activity, okay? And we have a bunch that actually activate the complex. So if we look at the type of molecules that deactivate, reduce the activity of the complex, we see that this general concept that I indicated previously is actually also applying to this protein complex, this enzyme complex, namely that molecules that signal an energy sufficient state have the tendency of deactivating regulatory enzymes that participate in catabolic reactions. So we shouldn't be surprised that ATP has an inhibitory effect. Acetyl-CoA, remember acetyl-CoA, energy-rich compound, okay? It's actually also the product of this reaction, has an inhibitory effect. NADH, the product of the citric acid cycle, is energy-rich, has an inhibitory effect. And so do fatty acids. So fatty acids have high energy content. If we have lots of fatty acids, we prefer to break down fatty acids for energy production. Carbohydrates are, let's say, not as energy rich and not necessarily as abundant usually. Depends. Okay. Now on the other hand, we see a couple of molecules that have an activating effect on the pyruvate dehydrogenase complex. AMP, okay, not energy rich, okay, poor in energy. The unacetylated coenzyme A, the free coenzyme A, and also the oxidized form of nicotine amide adenine dinucleotide, NAD+, all have an activating effect. It makes sense because they are poor in energy. Here and also later when we talk about regulation within the cycle, we'll see calcium appear also as an activator. Now calcium appears to be particularly important in muscle cells. So if we have an increase in intracellular muscle calcium concentrations, means that our muscles have to do work, and we need energy for that. And for that reason, it makes sense that calcium has some activating effect. Okay, so these are the allosteric effectors that affect the activity of the pyruvate dehydrogenase complex. Now let's talk about the transient covalent modification. It actually occurs here, appropriate, I think, a serine residue here in the E1 activity, which we can call pyruvate dehydrogenase, and we see that the unphosphorylated protein is actually the active form of the protein, shown in green here. And then if we phosphorylate this activity here, we inhibit not only the pyruvate dehydrogenase activity, but overall the whole complex, of course, because if we don't perform the first steps in this conversion of pyruvate to acetyl-CoA, then we can do the reaction. This phosphorylation is performed by a kinase that we can call pyruvate dehydrogenase kinase, and it's also subject to a bunch of allosteric modulators. So ADP and pyruvate have an inhibitory effect, NADH and acetyl-CoA an activating complex. But then, in order to regenerate the activity of the pyruvate dehydrogenase, a phosphatase can cleave off the phosphate, so pyruvate dehydrogenase phosphatase, and this can be activated actually also by calcium. So, transient covalent modification in the form of a phosphorylation is a relatively common theme in modulation of enzyme activity. So we talked about this tandem protein, for example. Here we have another example where phosphorylation actually has an effect on the activity of an enzyme. Here it's the pyruvate dehydrogenase complex. Now if we look within the citric acid cycle, we have to remember that regulation of enzymes is related to energy and the irreversibility of the reactions they catalyze. So we shouldn't be surprised that these three enzymes are actually our candidates here: citrate synthase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase complex, because all of these catalyze irreversible hexagonic reactions, and all of these are subject to regulation, as shown on this slide. Okay, I don't want to go into too much detail, but I want to remind you again of this general principle that energy molecules that indicate an energy-rich status, energy-sufficient status of the cell, have an inhibitory effect on catabolic reactions, and molecules that are poor in energy have an activating effect. So if we look at here within the cycle, citrate synthase, NADH, succinyl-CoA, citrate, and ATP all have inhibitory effect, while ADP has an activating effect. Now, we talked about NADH and ATP before. It's clear that they are energy-rich. Succinyl-CoA, I told you, has this thioester bond, is energy-rich, can be utilized for the production of an ATP equivalent within the cycle. It's also one of the products of the cycle, so say we have a negative feedback inhibition here. When the product of the cycle, actually one of the products, increases in concentration, we can slow down the pathway. With citrate, it's not that obvious. I may have mentioned this before, but citrate actually is a hidden form of acetyl-CoA. I'll talk about this more next time when we talk about the amphibolic nature of the citric acid cycle. But citrate is pretty much the transport form of acetyl-CoA across the inner mitochondrial membrane. But again, I'll talk about this more next time. So it's also energy-rich. Okay. Then isocitrate is inhibited by ATP, quite clear. It's activated by calcium, again, particularly in muscle cells, and by ADP. So whenever we have the accumulation of ADP or AMP, it signals that, okay, we need more energy, we need more ATP. And then alpha-ketoglutarate dehydrogenase is inhibited by succinyl-CoA and NADH and activated by calcium. Okay. So next time we'll talk about the amphibolic nature of citric acid cycle, and I'll explain what intermediates of the cycle can actually be utilized for biosynthetic reactions. At this point, I'm asking you to put away all your recording devices, all your computers. You may have a piece of paper, but turn off all